introduce what biosensor is. There have been many different definitions of biosensors. Sometimes any biomedical sensor, people call it also as biosensors. While for my personal definition, that is not true. So biosensors is a sensor where you have where you have a biological entity to as an element of the sensor. Okay, so biosensor by definition, or well, not only my definition, but in many other people, biosensor scientists, definition is this is a sensor which uh, which includes biological entity as part of its transducer, as part of its transducer. As part of its transducer. So the question is, why would we need to have a biological entity to be included as part of the transducer in a biosensor? Yeah, the reason is because we would like to that we would like to use that sensor to detect a biomolecular signal, either chemical signal or biochemical signal, which cannot be detected using physical sensor. So before the chemical sensor chapter, usually we discuss or we have studied many different types of physical sensors. Some sensors detect temperature, some sensors detect radiation or light, some sensors detect acceleration, some sensors detect the mass, some sensors detect the pressure and strain, other sensors detect the electrical field or those things. Some sensors detect magnetic field, and many different types of sensor which can be said about them that they detect physical domain signal. Well, in real life, especially when it relates to biomedical engineering application, many of the signal that we want to detect, that we want to measure, they are of biological signal. They are of biological domain, or they are of chemical domain, or they are of biochemical domain. For example, the content of, let's say, glucose. This is some common biosensor in the market already. The content of glucose in the blood. So you cannot use, for example, let's say, acceleration sensor to measure it directly. Okay, because it is not a physical it's not a physical signal. It is a chemical domain or biochemical domain. If you just want to detect whether glucose exists or not exists in your blood, we call it a qualitative biomolecule signal. A qualitative biomolecule signal, yes or no. But then if we want to increase the measurement, not only to detect the presence of glucose in the biomolecule in the blood, but also to measure how much quantity of that glucose in the blood, then we call it as a quantitative measurement of the biochemical quantity. Yeah, and this is just one simple example. Other example, like now, is there any SARS-CoV-2 virus in our body or not? This is a qualitative, physical, qualitative biological measurement also. And then, how many of this viral entity is there inside our, for example, inside our mouth or inside our lung? And this is again a quantitative biological measurement. Although, we do not deny that some of this biochemical signal can be detected using physical sensor. For example, like what? Glucose, again, glucose quantity in blood. There has been some research also to develop a biosensor. Well, it is, I can call it is not, I can say that it is not a biosensor anymore, but rather just a common biomedical sensor that uses infrared or red wavelength light, similar to pulse oximetry, similar to pulse oximetry used to measure the content of oxygen in our blood, that uses radiation to transmit into the blood and then measure the transmitted or the reflected wavelength. Okay, if it were pulse oximetry, usually it will measure then the entity, the wavelength region within red and infrared. There has been also some research to detect the glucose content from the spectral content in spectral of the wavelength of such blood transmitted or reflected light from blood sample directly from, for example, from our finger or from our vein, for example. And since every chemical usually has its own unique characteristic spectral absorption, then we can measure the reflectance or the transmittance of the light, that is irradiated into the blood to know the content of the glucose, for example. So this is some recent new development actually. It is possible, in the past, it is also possible, but usually it requires an expensive setup. With a very large setup, you take the sample from the blood and then you put it inside cuvette, and then you do UV spectral characteristic. You do also maybe Raman laser spectrometry or any other type of spectrometry, infrared spectrometry, and then you see the characteristic wavelength spectrum. From there, you know whether it contains glucose or not. If yes, how much of the glucose content is there contained in the blood. But what we mean with biosensor is, of course, a device that can be made portable and will be used on the field or at the point of care, we call it. This is point of care device. Point of care device means that the device is not used at the lab, but rather it is used at the clinics. So in many of the current clinical lab, the sample is taken and then diagnosed in the lab, biomedical lab. After that, the result is given back to the doctor for analysis. And there is a tendency maybe in the future that more of this instrumentation will be made more portable, more low cost, such that they can be put into the general practitioner clinics, or puskesmas or small clinics. And this is what we call as point of care clinics, point of care detection. So similar like what stethoscope have been now. Okay, so coming back to the biosensor. So only recently that those type of physical sensor based on radiation can be made small using a very small fiber optic, for example, a very small photo transistor or photo using LED. LED can now cover many different wavelengths from ultraviolet even until near infrared. And then you have the photodiode to detect the wavelength as well, having also wide, broad wavelength coverage. Then based on that, you can see at which wavelength that the characteristic wavelength of the transmitted or reflected light from your blood. From there, then you can estimate whether, for example, glucose exists there and how much of the glucose is there. But yes, this is a new development in biomedical sensor. This is actually, to my definition, it doesn't belong to biosensor definition, biosensor type, because it does not include any biological entity as its transducer. So the main definition is where, for example, when the sensor includes a what we call includes an enzyme to detect the biological entity, to detect a biochemical signal. Then we call it as a biosensor. So for example, so when we have a signal, yeah, and then we have different block. Yeah. The first one will be the biosignal entity, the biological entity. And when the biochemical signal or biochemical whatever it is, yeah, for example, in the form of molecules, yeah, or proteins or cell, interact with the biological entity in the sensor, it will create a certain physical signal, yeah, in the form of heat, in the form of electron transfer, etc. There is then we can attach a physical transducer, yeah, physical transducer to detect the physical domain change in the biological entity. Physical transducer. Yeah. Then the rest will be the same. And the rest, after that, it will be the same. We have an electronic signal processing. Okay. After that, it could be either displayed, yeah, or recorded or transmitted. Okay. Yeah. So display, record on memory, yeah, or transmit in the telemedicine, for example. Yeah, so this is what makes biosensor unique, because the transducer does not consist of a single transducer like what we used to have in physical domain sensor, but rather it has this couple between a biological entity and the physical transducer. Okay, so depending on which biochemical signal we want to measure, the biological entity also will vary. And depending on what type of physical signal that the biological entity change, then the physical transition could also vary. Yeah, for example, the biological entity here, what are types of the biological entity? It could be enzyme. So this is for, in the case of, for example, glucose detection. Glucose detection usually uses glucose oxidase. Yeah, for example, here I can give some example. Glucose sensing, then the biological entity would be GOD, glucose oxidase. Okay, glucose oxidase. But even recently now, there's a new development, yeah, in the biosensor world or in the sensor world. They have also non-enzymatic material recognition. Yeah, so for example, metal organic framework, MOF. So later you Google yourself. Yeah. So this is a non-organic material, that can be designed, yeah, to mimic the function of biological entity. So for example, it can mimic the function of enzyme to do catalysis of glucose into glucagon. So similar to oxidize glucose into glucagon, similar like GOD does, glucose oxidase does. Okay. So this is biological entity. However, yeah, it could also be not only enzyme, it could also be antibody. antibody, or antigen, depends which one we want to detect. Yeah, for example, in the if we want to detect SARS-CoV-2 virus, for example, like in the antigen kit, what they have is antibody that is specific to a SARS-CoV-2 virus. SARS-CoV-2 virus specific antibody being immobilized on that paper. Yeah, being immobilized on the paper kit. And then when they react, they have the enzyme, when they have the buffer, right? The flush and the buffer also contain secondary antibody that is bound to an enzyme. So usually it has this first antibody immobilized on the paper, and then you have the SARS-CoV-2 virus, for example, here attached to it. Then you have a secondary antibody that is bind also to this, but from the other side. And then usually the secondary antibody will contain an enzyme that will change the color of that region. Yeah, so this is why we see color change, colorimetric change. On the other hand, if we want to detect antibody, then we have to attach antigen. Yeah, you understand? So, for example, in the previous time at the onset of the pandemic, people use this antibody detection. They detect the presence of IgG and IgM. So detecting IgG and IgM that will be usually expressed in much more quantity when there is an infection that happens in our body. So in this case, then they could use actually different biological entity to detect the IgM and IgG in the blood. They can use, what they often use is antibody that is specific towards this human IgM and human IgG, or otherwise some antigen that can also bind to IgM and IgG. Okay, so for example, now if you want to detect whether in your body has developed an antibody, yeah, towards SARS-CoV-2 virus, yeah, like now they also develop this kind of biosensor. We can immobilize SARS-CoV-2 virus itself, the IgG, the antigen, you know, so to the biosensor, yeah, to the biosensor, so such that then when in the blood sample it contains enough antibody, it will bind to this antigen, to the SARS-CoV-2 virus. Then you can have a secondary antibody that bind to this, so that then the signal can be read. Okay. So yeah, biological entity can be anything that is of biological world. But as I mentioned, in recent days, there has been development in the fabrication, in the synthesis of MOF, metal organic framework, and other biologically biomimetic material that can function similar like antibody or like enzyme. Yeah. So what is the difference then? When to use enzyme, when to use antibody or antigen? Enzyme is a catalyst basically. Okay. Enzyme is a catalyst, while antibody and antigen, they are biological recognition molecule. So in the biochemistry, I hope you already learned in biochemistry that enzyme does not bind permanently to the substrate. Okay. So for example, if we have enzyme, then the substrate will bind, for example, like this. Then afterwards, it will create a product. So it will create, for example, with external like this. Yeah. So then the product afterwards will be detached from the enzyme, and the enzyme will remain there to react with other substrate. So similar like GOD, glucose oxidase, it binds glucose. The glucose is bound to the glucose oxidase. Then it is oxidized, react with oxygen. The glucose becomes glucagon. It will detach from glucose oxidase. and the glucose oxidaser is ready to bind with another glucose. Yeah, so upon this reaction catalysis, usually it will create also, as a result, in the case of glucose oxidaser, it will create H2O2, hydrogen peroxide, yeah, as a side product. So the H2O2, if then we also have horseradish peroxidase, if we have peroxidase enzyme, then that peroxidase enzyme can react with the H2O2. And there is a transfer of electron there during this reaction. And this transfer of electron, we can detect as using electrode. Yeah. So that's why as a physical transducer, the example also can be many depending on what physical changes occur at the biological entity that has undergone reaction with the biomolecule or with or others, yeah, that give the biochemical signal. So if it were electronic exchange, then we can use electrode to detect, such as in the case of GOD, yeah, glucose oxidase detecting glucose. Usually they are immobilized in electrodes, and the electrodes we use to detect the electrons being transferred during the catalytic reaction. What about if it were antibody and antigen? So antibody antigen, as we have drawn also here on the right side of this whiteboard, it will bind. Yeah, it will bind quite strong with the antigen. For example, with this antigen, and very difficult usually to detach from, to make them separate unless you flush them with uh, with a specific buffer to make the binding loose. Yeah, so this is related with the Ka, yeah, the association constant between the two. In the enzymatic reaction, the Ka and the Kd, the associative constant and the dissociative constant is more or less the same, yeah, more or less similar. So it can be reversible. The reaction can be reversible. In antibody and antigen reaction, the binding is very strong. So, and usually there is not much of electronic exchange. Yeah. So that is why we cannot use electrode to detect antibody and antigen reaction unless we modify the reaction. And what can be used then, what happens then when antigen is bound to antibody is mass change. Okay, so when this is being immobilized, yeah, on top of the substrate, yeah, initially it will feel only the mass of the antibody. So upon the binding of the antigen, of course, the mass on the surface of the substrate will change. Okay, so we can use any transducers that can detect mass change. What are their example? Yeah, for example, QCM, quartz crystal microbalance. We can also use SAW, surface acoustic wave. QCM is basically a type of piezoelectric, remember? The type of piezoelectric sensor. SAW is also piezoelectric sensor. Yeah? Only the difference is in QCM, usually it measures the resonance frequency at the bulk of the substrate of the sensor, while SAW measure only on the surface. And sometimes antibody or antigen or enzyme we can modify with certain colorimetric color, yeah, label, we call it label, colorimetric label, such that the color changes. So if the color changes, then we can detect also using radiation sensor. Yeah, radiation sensor, for example, photodiode in combination with LED, for example, then that can detect the color change in the surface of the sensor. Yeah, so there are many different transducer that can be used for this, and as we know, as we have learned from previous many chapters ago, that either if you use electrodes or QCM or KCW or radiation or photodiode, yeah, for example, and this all will produce electronic signal. And then you can just step them using amplifier, amplify the signal and put into analog to digital converter and put into microprocessor or microcontroller, and you can display or you can record in memory or you can transmit, yeah, to other devices or to cloud. So this is how the biosensor works. So the main key here is this, yeah, is this coupling of two different types of transducers. The first one is a biological entity, and the second one is a physical transducer. It is also perhaps worth to note that besides enzyme and antibody or antigen, we can also have larger biological entity as the biological sensing, yeah, part. It could be, for example, we can use cell. Cell, when it is cultured, we can, as probably also we have discussed in the previous chapter about array, microelectrode array. In one of the example given there in the book is that we can culture neuron cell on top of electrode and use it as a sensor to work, for example, neurotoxin, neurotoxin to as a screening to know whether certain chemical has toxic effect towards neural cells. Yeah, so in this case, then cell can be used as the biological entity, and the physical transducer will be electrode. Why we can use electrode if it is in the case of neuronal cell? Because of course in neuronal cell there has always been ionic transfer, yeah, between the inside and the outside of the membrane. Potassium, calcium, what else? Chlorine ions are being always exchanged across the membrane. We can also have tissue. Yeah, this is also not yet common. Tissue. Yeah, tissue, the whole like organ on a chip, yeah, which has been presented maybe last week by one of the group. Tissue or organ in a chip, and then use it as a drug screening method. Yeah, drug screening is basically also a kind of biosensor, yeah, where we test whether a drug will have a certain effect to the tissue or not, yeah, to the organ or not, and then detected the changes, physical changes in the tissue is being detected. Either you can detect using electrode or electrochemistry, QCM, and or radiation. Is there any question so far regarding this definition and concept of biosensor? Usually this will appear, yeah, in one of the questions for officer for oral final exam. And your batch is among the last batch to have oral final exam. So you have to be able to know, to understand and explain this concept. Any question? Grace, Anjali, Andy, Ezra?
No, sir.
No. Why so weak?
Kenapa, sir?
Okay, if there is no questions, we move forward to the textbook here. Yeah, so here, when they are enzyme, yeah, biosensor and microorganism, they are usually called as catalytic biosensor because catalytic reaction occurs there. In catalytic, remember that the catalyst itself will remain after the substrate is supported or is accelerated to make into product, then it will detach again, and the catalyst can react with the other substrate. That is why, as catalytic biosensor, they are reversible. They are reversible. Then we have another type, which is called as affinity biosensor. which is based on binding reaction, binding and non-binding reaction. And this is usually antibody, or specifically they are called as immunosensor. We can also use nucleic acid biosensor, and this is not yet explained in my previous whiteboard. And we can use DNA, either as single strand, and that will then it can detect the another single strand specific to its sequence. It's called as a complementary sequence binding. And we can have also a receptor and the ion channel sensor, because from the early chapter in this course that there are many receptors and they bind specifically to specific molecule, within our cell, in the surface, in the membrane of our cell, as well as also ion channels. Then we also have cell and tissue biosensor. This I have explained a little bit. So when we use the sensor to measure the metabolism of cell, impedance or electrophysiology, so we use cells or tissue as the biological entity. Then we will also here we cover the the other sense, different new technology like microarrays and nanomaterials for various sensing application by sensor. Maybe not yet in this book is the development of new materials like metal organic framework, MOF, to mimic the function of enzyme. Okay. So the first biosensor developed is, you can say, it is the here Prof. Clark, Prof. Leland Clark, oxygen electrode, and then he used it to detect glucose sensor using a dialysis membrane. And with an biosensor with integrated enzyme. So this is the component of biosensor, which I have explained in the previous whiteboard, that you have the analyte that is of interest that you want to detect. If it is just qualitative, then you just want to detect its presence or not present. But if you want to measure it, then you have to do quantitative analysis. You have to be able to also draw the calibration curve and then relate the measurement result with the quantity. Okay. So then the transducer itself always consists of two elements: a bioelement or biological entity, which can be enzyme. It could also be a whole microorganism. It could be also antibody. It can be also nucleic acids or cells or tissue, coupled with the transducer. So the function of the transducer here is to convert any physical changes related with the binding or reaction between the analyte and the bioelement into electrical signal. That is the function of this transducer. The bioelement itself, as I have explained, it could be enzyme, it is a catalytic reaction which will provide the catalytic reaction. It can be antibody or nucleic acid, which will be affinity sensor. It could be receptor on ion channel itself, or cells and tissue. Now it is more than that. So more than that. So for example, we have also PNA. Anyone ever know? This PNA is peptide nucleic acid. Peptide nucleic acid. So instead of made from deoxyribo, a molecule, like in DNA or RNA, ribonucleic acid, the nucleic acid is combined with peptide. We can also use peptide alone. Peptide, and then with peptide is basically a combination, a chain of nucleic acid. Amino acid, not nucleic acid. So amino acid. Peptide is combination of amino acid, and the combination of peptide will become protein. So then, based on whatever physical changes happen, we can detect the potential changes. For example, when there is electron transfer or when there is also electron flow, we can detect the electric current. We can detect the conductance. We can detect electrical impedance. Like now, what Elga is doing, your senior is trying to develop is developing a biosensor using electrical impedance analysis, electrical impedance spectroscopy. Okay, so this four can use electrode as the material for the sensor, as the material of the sensing element. And we can detect intensity and phase of electromagnetic radiation. Also, this can use electrodes. And we can detect mass if there is, for example, in this affinity sensor, affinity biosensor, we can detect the mass change due to the binding reaction. And this we can use QCM. We can use cantilever-based sensor. We can use surface acoustic wave, transducer, etc. And sometimes also when there is a reaction with this bioelement with the corresponding analyte, there might be thermal change. Temperature is being exchanged as well. In that case, you can use temperature sensor. You can use thermistor or semiconductor sensor to detect that minute thermal changes. Okay. So, what makes biosensor special? Yeah, because these bioelements usually they only react with specific molecules. Enzyme can only catalyze specific reaction, yeah, related to the substrate to make into a particular product. Antibody can also bind to specific antigen. Nucleic acid binds only to the other nucleic acid having complementary sequences. So they cannot bind to any just arbitrary molecule. They bind specifically to uh, the specific molecules. It's like lock and key concept, yeah, in biochemistry. It means then, from chemical sensor point of view, we have learned about selectivity and specificity, right? So biosensors are specific, very selective and specific sensors because of the high specificity of biomolecules and biological system with respect to the intermolecular interaction of interest. And that's why this can be exploited in biosensor devices if only there is highly efficient coupling between the biological and the transducer component. That's why one of the key, yeah, if we can come back to the whiteboard, yeah, one of the key to biosensors is how to couple this biological entity with the physical transducer. And this is usually in biosensor world, it is called as the process of immobilization. Immobilization. Yeah, usually these biological entity are also used, yeah, in biochemical analysis, but in the three-dimensional analysis inside a liquid, inside a chemical solution. And then you measure the reaction using UV spectrometry, for example. But in biosensor, we measure the interaction on the surface of the physical transducer. So that biological entity has to be attached, yeah, like this, yeah, attached, yeah, nicely on the surface of the physical transducer. This is a process called as immobilization. And immobilization is the key in any biosensor development. Okay, and there are four possible ways in the this one. Yeah, there are four possible ways. Four possible ways of immobilizing the biosensor on top of the surface. Yeah, what are they? They are this membrane entrapment, physical adsorption, then matrix entrapment, and then covalent bonding. Okay, covalent bonding. Okay, so let's pause it until this point. We will continue further the discussion about the different immobilization schemes to attach the biomolecules in the vicinity of the physical transducer. This